Original research article. Front. Psychiatry, August 12, Secretary, Volume. This article, From Default Mode Network to the Basal Configuration, Sex Differences in the Resting State Brain Connectivity as a Function of Age and Their Clinical Correlates. Zhan D. Conrin 1 Liang Zhan 2, 3 Zachery D. Morrissey 1 Mengqi Xing 1, for Angus Forbes 5 Pauline Maki 1 Mohammed R, MiladMohammed AjiloreOlusola A, Langenecker 1 Alex D, Leow 1, 4 *. 1 Department of Psychiatry, University of Illinois at Chicago, Illinois, United States. 2 Department of Electrical and Computer Engineering, University of Pittsburgh, Pennsylvania, United States. 3 Department of Engineering and Technology, University of Wisconsin-Stout, Menomonie, Wisconsin, United States. 4 Department of Bioengineering, University of Illinois at Chicago, Illinois, United States. 5 Department of Computational Media, University of California, Santa Cruz, California, United States. Connectomics is a framework that models brain structure and function interconnectivity as a network, rather than narrowly focusing on select regions of interest. MRI-derived connectomes can be structural, usually based on diffusion-weighted MR imaging, or functional, usually formed by examining fMRI blood oxygen level dependent, BOLD, signal correlations. Recently, we developed a novel method for assessing the hierarchical modularity of functional brain networks, the probability associated community estimation, PACE. PACE uniquely permits a dual formulation, thus yielding equivalent connectome modular structure regardless of whether positive or negative edges are considered. This method was rigorously validated using the 1000 Functional Connectomes Project Dataset, F1000, RRID, SCR_005361-1, and the Human Connectome Project, HCP, RRID, SCR_006942-2, 3, and we reported novel sex differences in resting state connectivity not previously reported. 4. This study further examines sex differences in regard to hierarchical modularity as a function of age and clinical correlates, with findings supporting a basal configuration framework as a more nuanced and dynamic way of conceptualizing the resting state connectome that is modulated by both age and sex. Our results showed that differences in connectivity between men and women in the 22 to 25 age range were not significantly different. However, these same non-significant differences attain significance in both the 26 to 30 age group. P equals 0.003, and the 31 to 35 age group, P less than 0.001. At the most global level, areas of diverging sex difference include parts of the prefrontal cortex and the temporal lobe, amygdala, hippocampus, inferior parietal lobule, posterior cingulate, and precuneus. Further, we identified statistically different self-reported summary scores of inattention, hyperactivity, and anxiety problems between men and women. These self-reports additionally divergently interact with age and the basal configuration between sexes. Introduction. In efforts to better understand the human connectome, various approaches have been used to identify and measure the modularity of brain connectivity. In these efforts, the brain is generally divided into a collection of communities or modules. Frequently, these modules can be subdivided into submodules, which then demonstrate hierarchical modularity and near decomposability, the autonomy of modules from one another. 5. Modules and submodules are comprised of a series of nodes with tight interconnectivity, whereas nodes of different modules have lesser connectivity. The connections between nodes are referred to as edges and can be either positive or negative in fMRI connectomics. A positive edge indicates that the activity in one node is positively correlated with that in the connected node, whereas a negative edge indicates the presence of an inverse relationship between the two. 6. When these concepts are applied to fMRI-derived networks, network organization identifies functionally related or coupled regions. The complexity and volume of data associated with large networks is enormous, and thus much work has been done to develop algorithms that better characterize and measure modularity. 7. In the popular approach of maximizing the Q-modularity metric, which is an epi-hard problem, typically the speed of computation comes with a compromise. 8. For example, the very efficient fast unfolding method does not guarantee global optimization and yields of a varying number of communities with each run. 9. Another area of variations is how positive and negative edges are accounted for. More frequently, the focus of computation is based on the recognition and measure of positive edges, as this is fitting for many types of networks. 10, 11. However, in measuring brain connectivity through fMRI networks, which usually focuses on fMRI blood oxygen level dependent, bold, signal correlations, positive and negative edges coexist. Most published studies have employed variable approaches of ignoring, thresholding, binarizing, or arbitrary downweighting to account for these negative edges, 12 to 14. Although quite different in their approaches, the common similarity is that the data involving negative edges is in some degree heuristically accounted for. To better address negative edges, we recently developed and published a novel method for assessing the modularity of functional brain networks, the probability associated community estimation, PACE, 4. Most importantly, PACE permits a dual formulation, thus yielding equivalent connectome modular structure regardless of whether one considers positive or negative edges. By exploiting how frequent bold signal correlation between two regions is positive versus negative, the edge, positivity, and negativity. This method was rigorously validated using resting state fMRI data from the 1000 functional connectomes or F1000 project dataset, F1000, RRID, SCR_005361-1, and the Human Connectome Project, HCP, RRID, SCR_006942-2, 3, and we demonstrated that negative correlations alone are sufficient in understanding resting state connectome modularity. Further, we explored whether our approach might be useful in the study of sex-based differences in healthy brain function, with the understanding that this might contribute to the discussion of how and why men and women differ in their expression of mental illness both in prevalence and type. When compared to various existing Q maximization-based formulations applied to the same two datasets, PACE yielded results that are both consistent with existing methods yet more stable and reproducible than alternative methods. Moreover, as a result of its superior reproducibility, and thus robustness, PACE was able to detect novel subtle sex differences in resting state connectivity that were not previously reported with Q-based methods. 4. These differences are conceptualized to be the end product of sexual development through hormones, socialization, and specialization. Given this and our understanding that brain development may extend into adulthood, in this study, we further examined sex differences in resting state connectome as a function of age. 15, 16. In this study, we comprehensively explored how these differences relate to parcellation resolution as it regards to the validity of these differences. Further, as secondary analyses, we investigated how PACE-derived modularity during the resting state may relate to self-reports of common psychopathology traits in relation to mood and anxiety in terms of the sexes. Last, we then proposed a more nuanced framework of conceptualizing the resting state human connectome, termed the Basal configuration framework, that generalizes and broadens the narrowly defined and perhaps more restrictive concept of default mode network. This new framework would allow us to better capture the complex dynamic interrelationship between different brain regions at rest that is further modulated by both age and sex. Methodology. Data. The data we used in this study is 811 subjects' resting state fMRI connectome data from the Human Connectome Project, released in December 2015, named as HCP900 parcellation plus time series plus netmats, https://dbead humanconnectome.org/data/projects/HCP_900. 3 different spatial dimensions of brain networks are explored: 100 by 100, 200 by 200, and 300 by 300, all derived using independent component analysis, or ICA. For study design, recruitment, and enrollment, as well as human subject consent and protection, please refer to 3. For details of the dataset and the procedure for connectome construction, please refer to HCP's official website and respective references 3, 17, 18. The study subjects' demographics are shown in Table 1. Table 1. Table 1. Participant demographics. Community estimation. In this study, we adopted the probability-associated community estimation, PACE, for framework to extract the hierarchical modularity of the resting state functional connectome, FC. Mathematically represented as an undirected graph FC, V, E, where V is a set of nodes, e.g., ROIs, and E the set of edges, and given a collection of functional connectomes S on V for each edge EI. J and E PACE considers the probability pair. 1. The probability of observing a co-activating relationship between node I and node J and S, i.e., the tendency that the 2 regions are active at the same time, and 2. The probability of observing an anti-activating relationship between node I and node J and S. To estimate this probability pair for each edge, we simply use the ratio between the number of connectomes in S having a positive or negative correlation values and the total number of connectomes in S, naturally the P-. P+ pair satisfies the following relationship. Then, given C1, C2, Cn that collectively form an n-way partition of V, PACE denotes the mean intercommunity edge positivity or negativity for the nth community Cn as. Here Cn denotes the number of nodes in Cn. Similarly, the mean intercommunity edge positivity and negativity between communities Cn and Cm are defined as. The PACE modularity is then the partition of V, C1 union C2 union Cn equals V, Ci intersection Cj equals empty set for all I is not equal to J. That maximizes the following benefit function psi that computes the difference between mean intercommunity and mean intracommunity edge negativity. Note, due to the elementary relationship between P- and P+, PACE can be equivalently thought of as maximizing the difference between the mean intracommunity and the mean intercommunity edge positivity. Constructing the PACE null model and testing the statistical significance of each bifurcation and between sexes. In our current implementation, a top-down hierarchically bifurcating tree is constructed. For each branch at a specific hierarchical level, PACE further splits that branch into 2 subsequent groups by maximizing psi with respect to that level using simulated annealing 1920. Then, a non-parametric procedure is used to determine the level of statistical significance for such a split. Note that by stopping a branch from further splitting when there is no evidence in support of this bifurcation, PACE can in theory yield any number of communities, i.e., not restricted to powers of 2. This non-parametric procedure leverages the interchangeability of edge positivity/negativity between any 2 edges under the null distribution given the observed data. Since under null there exists no connectome modularity, and thus 2 edges can be randomly selected and exchanged. Thus, assuming there are no modular patterns of co-/anti activation, we could sample the distribution of the PACE benefit function psi by first randomly choosing 2 edges and exchanging their edge negativity/positivity probability pairs. Randomization is iterated over the entire connectome, followed by recomputing the PACE trees by maximizing psi with reshuffled edges. Then, this entire process is repeated for 1,000 times, yielding 1,000 samples of psi under null. Last, to determine the significance of each split, the actual psi achieved by the original data is compared to the 1,000 sampled psi values under null at the same pace level, if the former lies within the top 5% of the latter. Such a split is determined to be significant. P less than 0.05. Figure 1. Figure 1. Figure 1. Testing the statistical significance of each bifurcation for the PACE hierarchical modularity tree. First, we derive the community structure from the observed data by maximizing psi, defined as the difference between mean intercommunity edge negativity and mean intracommunity edge negativity. Then under null, we assume edge interchangeability, thus allowing us to sample the null distribution of psi by randomly shuffling edges to create 1,000 permutations. For each permutation, we rerun PACE to obtain bifurcation trees and record the corresponding psi values, and conduct max pooling for level 2 and up, yielding the null distribution of psi for each level. Then the bifurcation of the original data at each split will be treated as significant only when the psi achieved by the original data lies within top 5% with respect to the null distribution. To test if PACE modularity differs between sexes, we employed a similar permutation testing approach. Specifically, by permuting with respect to sex, under null the sex of a person is interchangeable, we could recalculate the probability pairs at each edge specific to each sex, thus allowing us to generate samples of male/female hierarchical modularity under the null. The difference between sexes is then quantified using the normalized mutual information, NMI, computed between the two sex-specific PACE modular structures, and the actual realized sex difference is compared to the resampled differences, thus yielding a p-value indicating the level of statistical significance. Clinical correlates of connectome modularity relate systems-level PACE modular structure to subject-level characteristics. Firstly, in order to determine the statistical significance of the observed differences between sexes, a 10,000 permutation testing was performed within each age bracket. By first randomly reassigning each subject's sex and then recomputing PACE in order to control for multiple comparisons using the procedure described in section constructing the PACE null model and testing the statistical significance of each bifurcation and between sexes. Then, as part of the HCP data release for behavioral data, we downloaded and used, under the category of Psychiatric and Life Function 1, the already de-identified Achenbach Adult Self-Report, Syndrome Scales and DSM-Oriented Scale. Here, the Achenbach Adult Self-Report for ages 18 to 59, 21, was administered to obtain a broad variety of self-report psychiatric domains. Specifically, the 123 items from Section 8 were used to generate the ASR Syndrome Scales and the ASR DSM-Oriented Scales, and in this study, we primarily focused on adult self-report, ASR, DSM depressive, anxiety, inattention, and hyperactivity problems scores, Table 1, and tested if there are sex differences after controlling for age. In particular, using a general linear model incorporating an intercept, main effects, as well as a sex-age interaction term, the statistical significance of a sex effect is tested by centering age within the age range 22 to 35 across all subjects for all ASR scores. Separately, for each of the 3 resolutions available for HCP 100 ROI, 200 ROI, 300 ROI, we applied PACE to extract modularity, separately for each sex, as well as for the combined total sample, and determined the optimal level of bifurcation using the null model procedure introduced above, thus at the most global level, i.e., first level of PACE, yielding 2 modules. Operationally, as one of the 2 modules includes regions traditionally considered DMN, we will use the term PACE-derived task negative network, TNN. In the remainder of the paper, while the other module will be denoted the PACE-derived task positive network, TPN. To summarize the overall activity of TNN and TPN, for each individual we computed the average correlation value within TNN, average TNN, and TPN, average TPN. As secondary analyses and to validate our PACE-derived first-level modules, we explored behavioral correlates of TPN and TNN. We conducted partial correlations, controlling for age, between average TNN and average TPN and each of the 4 ASR-DSM problem scores. Previous studies have demonstrated cognitive and effective correlates of TPN and TNN activity associated with the same domains in the ASR-DSM scales, 22 to 25. Results: ASR-DSM problem scores. For ASR-DSM inattention and hyperactivity problems, scores men are significantly higher, assessed at mean age of 28.75 years. For inattention problems, men higher than women by 0.36 points. Standard error SE equals 0.17, P equals 0.031. For hyperactivity problems, men higher than women by 0.33 points. SE equals 0.026, P equals 0.0023. While women's self-reported ASR anxiety problem scores are significantly higher, assessed at mean age of 28.75 years, women higher than men by 0.83 points. SE equals 0.19, P equals 8.5 E06. In addition, overall, there are age effects for both anxiety and hyperactivity problem scores, lower scores as age progresses. For anxiety problems, beta equals -0.107, SE equals 0.034, P equals 0.0017. For hyperactivity problems, beta equals -0.080, SE equals 0.026, P equals 0.0023, but not for inattention problem score. For ASR-DSM depressive problems, there is a significant age effect. Beta equals -0.137, SE equals 0.045, P equals 0.0026, but not a significant sex difference. PACE modularity results. Across the entire sample, PACE-derived modularity at the most global level, yielding 2 modules operationally defined as the TPN in red and the TNN in green, is shown in Figure 2 for each sex and each of 3 parcellation resolutions, 100-, 200-, and 300 ROIs. As expected, sex differences are visually more easily appreciated for the higher resolutions. P-value of sex differences equals 0.0001 for 100 ROIs, and less than 0.0001 for both 200 and 300 ROIs. Figure 2. Figure 2. Sex differences in resting state modularity revealed using the probability associated community estimation, PACE. Slices obtained from 100 ROI, 200 ROI, and 300 ROI resolutions using the Human Connectome Project HCP data. At PACE Level 1, 2 brain modules are extracted, here shown as the red community, corresponding to task-positive network or TPN, and the green community, corresponding to task-negative community or TNN. Mixing of communities is shown in the overlay in yellow. Since HCP parcellation is ICA-based, components may overlap, thus resulting in the mixing of TPN and TNN. As expected, with increasing spatial resolution from 100 to 300 ROIs, sex differences also become more significant. For both 200 ROI and 300 ROI resolution P less than 0.0001, significant after Bonferroni correction with a p-value cutoff 0.05/3. Differences between the sexes, boxed, include precuneus, hippocampus, and amygdala. MNI coordinates x equals 12, y equals -14, z equals 22. Second, Figure 3 details the corresponding complete hierarchical modularity for men and women across all 3 age groups, visualized as bifurcation trees, after applying our null distribution procedure. See section constructing the PACE null model and testing the statistical significance of each bifurcation and between sexes. Interestingly, we identified an additional split in men during this procedure, resulting in 8 modules for women and 9 for men. Figure 3. Figure 3. A graphical representation of the PACE-derived male and female hierarchical modular structure, optimally determined using the proposed null model procedure. Female communities are displayed in the top row of each level with their male counterparts displayed on the bottom row. Axial and sagittal slices are shown for each community. Note that while female community bifurcation was no longer significant after PACE level 3, 8 total communities, a further significant bifurcation was observed in males. Third community from left at PACE level 3. P equals 0.006, yielding 9 total communities. MNI coordinates x equals 12, z equals 22. PACE modularity as a function of age. Next, we explore connectome modularity as a function of age, with results shown in Figure 4 where we visualize the sex-specific modularity in each of 3 age groups, 22 to 25, 26 to 30, and 31 to 35. Although sex differences did not reach statistical significance in the 22 to 25 years old age group, visual differences were noted in several areas. Sex differences then reached statistical significance in the 26 to 30 years old age group, P equals 0.003, and even more significant in the 31 to 35 age group, P less than 0.001. Interestingly, female modularity remains largely stable across the 3 age groups. A head-to-head comparison between the 22 and 25 years old group and the 31 to 35 years old group was indeed statistically not statistically significant, whereas males exhibit different patterns across age groups, particularly from the 26 to 30 to 31 to 35 age groups. Also, the areas of significant differences in men are in brain regions where transitions occur as they age, gradually from more probabilistically task-negative, green, into task-positive, red, including the precuneus. The inferior parietal lobule, the prefrontal cortex, the hippocampus, the amygdala, and the middle temporal gyrus. C. Supplemental video. Figure 4. Figure 4. Sex differences across 3 age groups, visualized at PACE level 122 to 25, M, 106, F, 70, 26 to 30, M, 152, F, 197, and 31 to 35, M, 106, F, 180, years old, CF Table 1, from the 300 ROI HCP data. Sex differences were not statistically significant in the 22 to 25 years old group, but reached statistical significance in the 26 to 30 years old group, P equals 0.003. Significant after Bonferroni correction at a p-value cutoff of 0.05/3, and is also statistically significant in the 31 to 35 group, P less than 0.001. Areas of significant differences are in brain regions where transitions occur from TNN, green, to TPN, red, in men but not in women, boxed areas, black, precuneus, blue, inferior parietal lobule, magenta, prefrontal cortex, orange, middle temporal gyrus. F equals female, M equals male. MNI coordinates x equals -48, y equals -62, z equals 22. Note, instead of indicating voxel-wise differences between sexes, the highlighted areas localize where modularity as a network property differs significantly. Secondary analyses. Correlation between PACE modularity and ASR-DSM problem scores. Secondary post-hoc partial correlation analyses, controlling for age, confirmed that average TPN is negatively correlated with ASR anxiety and inattention problem scores in men, but not in women. All other correlations are statistically non-significant. ART equals -0.131 and P equals 0.01 for anxiety problems. R equals minus 0.127 and P equals 0.015 for inattention problems, p-values uncorrected. Last, across the entire sample, average TPN also positively correlates with average TNN. R equals 0.37 and P equals 1.4 E minus 28. Figure 5. Figure 5. Figure 5. This figure plots the average Fisher's Z-transformed correlation within TNN, average TNN, against that computed within TPN, average TPN, across the entire sample. There is a highly significant positive correlation, R equals 0.37 and P equals 1.4 times 10 to the power of negative 28 between the two, suggesting a synergistic relationship between them than antagonistic. Discussion. In this study, using the F1000 and the HCP dataset, we comprehensively examined resting state connectome sex differences over the course of early adulthood. In particular, how they are modulated by age as well as their clinical correlates. Our findings lead to a more nuanced picture elucidating 1. How fMRI-derived brain connectivity exhibits both sex-specific configuration and age-dependent dynamic reconfiguration, the latter primarily in men, from early 20s to mid-30s, and 2. How this sex-by-age configuration during the resting state relates to self-reports of common psychopathology traits in otherwise healthy subjects. Although sex differences were not identified at a level of statistical significance in the 22 to 25 years old age group, a trend difference is observed in several areas in the brain. Specifically, in areas either designated as or functionally closely coupled with DMN, we identified a gradually shifting connectome configuration over time in men versus women, away from probabilistically more task negative and toward probabilistically more task positive. In this sense, our probabilistic framework frees up a brain region from having to be hard-parsed, e.g., DMN versus non-DMN, thus allowing functional brain units, i.e. modules, to work synergistically at times, and to act nearly decomposable at other times. Of interest, at the same time that basal configuration differences are identified with increasing statistical significance with age, the differences also show increasing statistical significance within individual age groups with increasing image resolution. What might contribute to the sex differences observed in the present study? Some studies have shown that hormones like estrogens and progesterone might modulate the resting state functional connectivity within the DMN 26-27. Further, in support of the argument that hormones play a role in connectivity differences, oral contraceptives taken by women are known to impact functional activation to emotional stimuli and resting state functional connectivity 26. 28, 29, while women on oral contraceptives may have smaller hippocampal volume, 30. The results of our study indicate that the differences between men and women do not appear statistically significant until we reach the 26 to 30 years old age range, and the trend appears to increase into the 31 to 35 years old age group. This is well past the age of puberty, and so the first question to arise is why do these differences gain statistical significance at an age range substantially older than the onset of puberty? One likely hypothesis is that men move to develop sexual dimorphism in function later in adulthood, perhaps through later development of white matter connections than women. Along these lines, one recent study suggested that the 20 to 25 years old age range may be a point of inversion of earlier sex differences in cortical thickness and folding differences. However, this study was limited in that they did not have a cohort extending into the 30s to fully probe this potential interaction between age and sex. 31. Given these sex-based age-dependent differences in otherwise healthy people, a natural further course of inquiry is to ask how these differences might relate to the known differences in the prevalence and symptom expression of mental illness between the sexes. Among clear differences between men and women in terms of mental health is that women are much more likely than men to experience episodes of depression, 32 to 35. Examples of frequently investigated factors are differences in hormonal development, societal roles, communication patterns, and ways of coping with stressors. However, the lack of definitive conclusions to date suggests that additional and unrecognized factors might be at play. 34. As such, it may be useful to consider whether sex-based differential brain network connectivity might be one of these unrecognized factors. Clearly, it is a complex picture, and it is important to note again that exposure to sex hormones is already known to affect brain network connectivity. One example of this is discussed by Ottowitz et al. That addition of estrogen to postmenopausal women is shown to increase connectivity between the hippocampus and the prefrontal cortex, which is part of the frontal limbic circuitry known to play important roles in depression. 36. While Mengqi et al. Provide another example in which they find that perimenopausal women exposed earlier to hormone replacement therapy perform better in various cognitive tasks than those who are exposed after a longer time untreated. 37. One area in which differential brain network connectivity possibly contributes to sex-based differences in depression is rumination. Rumination is the repetitive thinking and focus on negative mood states. 38. It is also a form of self-referential processing of information. 39. Areas identified as playing a role in self-referential processing have included medial prefrontal cortex, anterior and posterior cingulate cortex, insula, temporal pole, hippocampus, and amygdala. 32. 39 to 47. Note that in our current study, we identified sex-based differences in areas highly overlapping with these. Another major difference recognized in our study is in regard to self-reported inattention, hyperactivity, and anxiety problem scores. While depression problem scores are similar between the sexes, men have higher self-reported inattention and hyperactivity scores than women. Women, by contrast, have higher overall anxiety problem scores. Further, correlation analyses revealed that overall average TPN is positively correlated with average TNN, and in the male group, the former further inversely correlated with attention problems and anxiety problem scores. Last, note that our sex difference findings in self-reports are generally in line with Gur and Gur's paper, in which they found that men have earlier declines in frontotemporal areas associated with attention, inhibition, and memory. 48. Indeed, men tend to express symptoms of depression differently than women in terms of acting out behaviors such as substance abuse, restlessness, and suicide. 34, 49. While this difference in expression of depressive symptoms is not fully understood, it is possible that the tendency toward higher degrees of inattention and hyperactivity in otherwise healthy men at least partially explains why men tend to exhibit more externalizing behaviors when depressed. This finding is also interesting to consider in the context of Engelhalliker et al. Findings regarding sex-based connectivity differences in adolescence, in which the development of greater interhemispheric connectivity in females leads to better performance in attention tasks. 50. There are a few limitations in our current study. First, while PACE is a rigorous framework to model negative correlations and to pinpoint modular differences in a hierarchical fashion, it is only able to localize on a modular level where groups differ, but not on a voxel level. Second, the fact that we did not find any sex difference in the youngest age range may be simply due to a smaller sample size. Similarly, the fact that we only found basal configuration correlations with self-reports in the male group merits more discussion. While this can be due to a lack of power in the female group, we did find modestly robust correlations in the male group. Alternatively, as recent lines of evidence suggesting that in women cognition is a function of the menstrual cycle due to differential effects of estrogen and progesterone. 51. Another plausible explanation is that in women the basal configuration and self-reports such as ASR scores are also dependent on the menstrual cycle. If so, the lack of correlations in the female group is merely a consequence of the fact that the functional imaging data and the self-reports were obtained on different days and/or were differentially dependent on phases of the menstrual cycle. Future studies can use a repeated measures framework to estimate the relationship between change in the basal configuration in relation to these self-reports and other potential factors that may develop later or differently in males. Conclusion. In sum, this study contributes to a growing literature on the limitations of our current conceptualization of resting state networks. Our research supports the argument that conceptualizing the default mode network as being the network active at rest, thus driving resting state brain dynamics, while the activation of other networks is responsible for taking over during active tasks, is a concept that requires continued consideration. In time, it is possible that a more nuanced model of functioning will be identified as the leading theory on brain connectivity. In this fashion, we argue that the current perspective is an oversimplification of the actual complexity of brain connectivity, as on a subject level, with increasing default mode network activity, the functional activity in other networks, generally considered to be task-positive networks, increases as well. Taken in conjunction with previously published studies in which increasing connectivity is an important factor in overall functionality. 18. This study lays the foundation of a more nuanced framework for better conceptualizing the resting state. To this end, our results supported that 1. The basal configuration exhibits distinct sex-specific dynamics by mid-30s. 2. The basal configuration diverges during early adulthood between the sexes, in that there is globally an age-modulated reconfiguration primarily in men, but not women. 3. The basal configuration correlates with self-reported measures of personality traits, at least in men. 4. Whereas in women the basal configuration is further conjectured to likely exhibit a strong dependence on the menstrual cycle. Author contributions. All authors wrote the paper. LZ, ZM, MX, AL designed the study and conducted the calculations. AF, PM, MM, OA, and SL provided the discussion on the results. Funding. This work has been partially supported by NIH AG 056782 to LZ and AL. Conflict of interest statement. The authors declare that the research was conducted in the absence of any commercial or financial relationships that could be construed as a potential conflict of interest. Supplementary material. The supplementary material for this article can be found online at https://www.frontiersin.org/articles/10.3389/FPSYT 2018.